Hello class, this is Miss Evelyn, CNM Veterinary Technology Program, and this is the first opening recording for the Microbiology Part 1. You have a PowerPoint that is available to you that you can print out, save, pull up in your devices, and make your own notes. I'm going to try to cover this PowerPoint as a portion of lecture and be able to then reference it. You can reference it back on your own time schedule. So with that, I'm going to open up and share the files. And this ought to take care of it. So just one comment about the PowerPoint slides themselves. I have a Mac that I set it up on. I keep trying to work with the some of the areas of the margins and PowerPoint or the points, the little bullet points, and every time I save it, it kind of reverts to other things. So I don't think that there's anything cut off on this presentation, but I will mention if there is. So starting on Microbiology Part 1. In this, you're going to be covering basically into Unit 7, and in Unit 1, we're going to cover some of the general safety areas for laboratories. So for this Part 1, my first part that I just want to mention is that I do give credit for all of the microbiology PowerPoints to Miss Gina Miko. She actually taught the original portions of microbiology, pulled it together for us with the animal veterinary side approach to it and the necessary skills. And then I have actually kind of taken over her basic material. So I do continue to want to give her credit, Miss Jean Amico. No, that's not a test question, but I do give her credit for the basic outline. So this material, as far as microbiology is concerned, and again, it's Unit 7 in your textbook. This is not a full course in microbiology. If you love microbiology or you get a twinge for it, take a full course one time. Okay, so it's not in place of a complete term in microbiology. You will be demonstrating, we won't be demonstrating all of the techniques or organisms that might be available, and definitely you will not be a microbiologist after the next 2 to 3 weeks. Hopefully it will be very practical in its approach with an overview for general practice as you are later a registered veterinary technician. You may end up working at a practice that has a lab that actually does some microbiology in it, or you will be preparing specimens to go out to a diagnostic lab. But hopefully this will be very practical in those approaches. Introduces our basics, the basics about bacteriology and microbiology. You will be introduced to organisms next week. This week is all about the skills and basically kind of building the firm foundation. Eventually, you will be practicing your skills in the coursework, and that would be there for general practice. And this will also introduce common laboratory diagnostics for microbiology. Our goals for the microbiology section is to introduce you to microbes, morphology. How do those look? What is their shape, their color? When they grow on a plate, how do they look? So morphology. And then with that, that leads to identification. You will be doing a skill eventually when we have the hands-on skill portion of what's called streak isolation. You will do a gram staining process. They'll be introduced to other basic tests and run some sensitivity tests. You will be definitely introduced to diseases and their organisms. You've already heard some of those in animal diseases last term. And then also into the fungi, the funguses, and dermatophytes or ringworm. And a little bit about vaginal and oral or ear identification. Again, very practical to the general office application, not for a full diagnostic laboratory situation, but general for office application. And to increase understanding to larger laboratory tests, understand proper collection and transport of your samples, and that the basic skills that you can do to gather information that may assist the veterinarian and lead to a diagnosis. Sometimes just doing a gram stain and being able to tell the doctor, I see a cocci, it's in clusters, and it is gram staining. It's a positive gram stain, purple in color, and the doctor may have a better choice for an antibiotic just based on that information. Okay, there are 14 learning objectives in microbiology. Some of these are outlined as learning objectives for this week, and some will be there again, doubled up again when you do this with the hands-on. But to actually follow microbiology safety and some general lab safety procedures, you will be having a deeper or more, I'd say complete, just a different angle of a safety presentation when we do the face-to-face. It will make a little more sense when we're in the classroom. Proper use of PPE goes along with that. Proper specimen, sterile techniques. Now, you may not be really dealing with sterile, but there are definitely techniques that you will be introduced to in order to have the best outcome of the tests that you do. How to prepare a slide for gram stain. When we do the hands-on skills, that will be one of your major skills to try to perfect in a short period of time, will be to actually do your gram staining. How to use your microscope properly and how to read that gram stain ID or organisms. And again, we go into organisms very thoroughly, albeit quickly, but thoroughly next week. How to perform a 4-quadrant streak isolation, how to label your plate properly. Understanding what a McFarland standard is, observing and measuring what's called zones of inhibition that goes along with the McFarland standard. Understanding hemolysis, when you have a blood agar plate and different organisms will actually do a different pattern of growth or absorption, or as I call it sometimes, eating up the blood in that agar. That's the hemolysis. And different patterns will help you to gain an understanding or potential identification for an organism. Observing and identifying various types of media that you will be introduced to, performing various tests, observing yeast and dermatophytes, and understanding what a dichotomous key is and how you can use that in order to identify organisms. Again, you can see that a lot of these are actually learning objectives with the verbs of doing something. And in this part of the course, in a lecture type, it will be to understand, gain the knowledge to be able to do the items, okay? So the lecture lab material will be your Serios textbook. It is the Laboratory Procedures for Veterinary Technicians, and it is this green and yellow textbook is the one that will be referenced totally. You will have read most of that textbook or all by the time you're done of this summer course with the 2 sections. This first week we're looking at Unit 1, pages 1 to 31, and then Unit 7. And it's all of Unit 7, but then in week 2 you will be coming back and using Unit 7 and the Appendix B to actually reference and understand some of the organisms that you'll be introduced to. The goal again for this summer is to gain the knowledge to form your foundation to actually perform your specific laboratory skills, and then how to accurately record those results. The hands-on skills will follow in a face-to-face laboratory. So you will have to remember, though, what you learn. We are not going to preface each laboratory with an hour or an hour and a half of lecture. That's what this 6-week section and next 6-week section in hematology is going to form the foundation. So you probably want to download the PowerPoints, download other presentation materials, and be able to reference them back, because of course once the course is done, you don't have the student access to the materials. So you probably want to plan ahead, build some files in your computers, and save the documents so you can come back to them in the next session when we have the hands-on. Well, the very first thing, this is in Unit 1, okay, Chapter 1 area, is safe lab procedures. The very foundation of working in a veterinary laboratory is to treat all specimens as if they're potentially pathogenic. So pathogenic means they can pass disease. You always want to think safety first. Watch where you walk. Don't trip over a cord. If somebody else has a inoculation loop in their hand and they're dealing with plating an organism onto a Petri dish area, you don't want to bang into their elbows. So watch where you walk. Think safety first. You're going to wear your lab coat, gloves, masks accordingly. And with COVID-19, you probably will wear your mask, and eye protection is necessary. But you will also remove that PPE, that personal protective equipment. Know what that abbreviation is, personal protective equipment, when you're going to exit the room. The proper order to remove your PPE is that you would first remove any gloves that you have on, then wash your hands, and then reach to unbutton and take off your lab coat, and then wash your hands again. The reason you do it in that process is you want to take off your gloves in a sterile, appropriate, clean manner. We will show that to you the first time in lab. But you don't want to start unbuttoning your lab coat before you take your gloves off, because then you're just putting any of those goodies that you're trying to keep away on your clothing. So you want to remove your gloves, and then you wash your hands, and then go and remove your lab coat, hang it up, put it on your chair, and then you're going to go and wash your hands again. So if you need to exit for personal relief needs or anything else, make sure you give yourself enough time to be doing that proper order of removing your PPE. You also don't want to touch your eyes or deal with contact lenses or rub your mouth, and no gum chewing, okay? The reason is, if you're sitting there gum chewing, because I think the video of me shows a little bit. If you do gum chewing, you can just imagine if there are microbes in the air, it might get on your gum, and then you're going to get infected. Bactericidal, okay, being able to kill bacteria, disinfectants can be used, and you're going to apply all of these items as best as possible when you're out in clinical practice. Now, different offices might have different protocols. Some may not want you to wear gloves every single time. You may need to be able to ask your employers or your clinical people to be able to, that you can put gloves on. It is becoming more and more of a standard of practice and should be done, okay? Especially if you're handling anything that you think may be potentially infectious, which brings us back to the top of the slide, that you're going to treat all specimens as being potentially pathogenic. Slide number 6. Chapter 1, continuing. Make sure that you understand, if you could recall it for a quiz, what OSHA stands for. OSHA, Occupational Safety and Health Administration. There is also OSHA as the Occupational Safety and Health Act, which is administered by the administration. But Occupational Safety and Health Administration. They regulate all of our safety standards, and if you ever have a visit from OSHA, you're going to be very nervous through the visit, because they will be looking very closely at many different kind of protocols. Equipment that might be present and included in both our lab area and in veterinary offices. You may have an eye wash station. We will show you make sure you know where that's at in our classroom. Fortunately, we don't really have too many chemicals or anything that we work with that can be very irritating to our eyes, but if you did get a spritz of something in there, you should go to the eye wash station. You may have a spill cleanup kit. sharps containers of various sizes. And there could be safety posters. MSDS, Material Safety Data Sheets. So know both of these abbreviations, OSHA and MSDS. I have a helicopter going overhead, so we're just going to let it roar here for a second. So MSDS, and you'll see in Chapter 1 where it talks about that the MSDS presents the information about any chemical or medical product or other item like that in a very standardized, user-friendly system, 16-section format. No, you do not need to know, for this class at least, what those 16 sections are, but do understand that it is very ordered, and you, as a veterinary technician, you may need to be referencing those and know where to look for different items. Bloodborne pathogen standard. Again, this goes back to the previous slide that you want to treat everything as potentially infectious, whether it is a fluid from an animal, blood, waste materials, urine or feces, if it is a tissue waste, you do want to treat everything as potentially infectious, and that is our general bloodborne pathogen standard. And then employers provide your necessary PPE, but the other part of that is employees must use or should use that supplied PPE. And then again, you really want to design your laboratory to enhance safety and efficiency. Okay, I'm not sure if I skipped one there. Okay, Chapter 2. Okay, so even though there's several quick little chapters in the first unit, first 31 pages, do highlight off of this PowerPoint some of these main points that I'm bringing out, but definitely read all of the pages. Read it over. Get the basic understanding of how it falls into place. So blood collection tubes. When we actually have some hands-on, you may be introduced then through, especially into hematology section, what those color-coded caps and additives are. We won't really be dealing with that in the microbiology or the parasitology or urinalysis section, but that statement actually will transfer over to the hematology course. There's what we call bench equipment and handheld equipment. There is a supplemental video, or probably broken apart into 1-minute sections of video, introducing you to the bench equipment that we have at C&M. These include various types of centrifuges, slide warmers, what they call the aliquot mixer, incubators, water baths or heat blocks, and of course chemistry analyzers. These are basically equipment that stay put on a countertop, a specific area, and I guess with this would also be microscopes. Handheld equipment may be something that's much smaller. It could be taken cage side. It could be taken into a clinical exam room and utilized there. Such things as a refractometer, you will be introduced to that. You will be introduced to various cell counters, pipettes, petri dishes, and in our area for microbiology, different media. Chapter 3, so moving along those first 31 pages, talking about our microscope. And I know that you've already used this, and you've used it in a couple previous classes, not only in veterinary technology, but probably in chemistry and biology. But at C&M, in our veterinary technology area, we have binocular compound light microscopes. So they're binocular. They have a double eyepiece, and you should utilize both eyepieces. Train your eyes to be able to look through them for the best longevity, best diagnostic purposes with that microscope. They are built to really give you a dual eye use, okay, versus a monocular type of microscope. You have a mechanical stage that helps hold the slide in place. You have coarse and fine adjustments. You have the ocular system, so the binocular eyepiece gives you 10 power, 10x, 10 power. Okay, 10 times the magnification just off of the eyepiece. Your objective system are your 4 lenses, adjustable lenses, they rotate into place. You have 4 power, 4x for scanning. 10x is what we call low power generally. 40x is termed your high dry, and then 100x is for oil immersion. And for microbiology, you will be utilizing the oil immersion, and also in hematology. Your general parasitology work and urinalysis quite often just goes as far as the high dry. You always want to support and carry your microscope with 2 hands, using lens paper to clean it. Kim wipes can also clean the stage and other body parts of your scope, cleaning oil off of the stage, and also very handy for cleaning up the backside of your glass slides before you're going to look at them to remove any staining debris. And always, always do firmly grasp the plug. Do not unplug just by a cord. You want to grasp the plug and pull gently and firmly there out of the outlet. There is also a supplemental series of, again, in about 1-minute sections on the microscope that will be you will have some lab work assignments with that. Chapter 4 talks about the metric system and calculations. Read the chapter well. There probably will not be much use for that in the microbiology section, but definitely when you get to parasites, knowing the view of your field, the field of view of your microscope, and what the sizes are is a very key factor to understanding what kind of parasite egg you may be looking at. And also with crystals in urinalysis. But you should know the abbreviations, that a meter is an M, gram is small g. If it's a liter, it's a small l or a capital L. And then there's a nice table in there on page 25 that talks about the basic units, deci, centi, milli, micro, okay? Dilutions, there's a little bit of a reminder about doing dilutions, and there are several times within laboratory work that you will be utilizing that. Understand your pH scale. Less than 7 is considered an acidic solution. The pH of 7 itself is usual neutral, term neutral, and above 7 is basic. And there is a help there in that chapter to remind you about temperature conversions as you are introduced also in your calculations course. 0 degrees Celsius or centigrade, either one, equals 32 degrees Fahrenheit. Quality control and record keeping. Again, this will make a lot more sense when you're actually doing the work with the hands-on, but the basic premise is garbage in, garbage out. If you don't do a good skill on your test, you will get bad results, inaccurate results, maybe, you know, totally wrong, false results, okay? So just remember that you want to perfect your techniques as they're presented in class. Take time to practice those, okay, when we have that hands-on time. Employ your attention to detail, and as we say, ATD, attention to detail. Be watching again your safety factors, be understanding what is the sample you're looking at, especially in parasites. That is a very critical factor, is where did this sample come from? And then to be able to seek accuracy, you're going to practice precision with your tasks that you do. Accuracy plus precision actually equals your reliability of the test. And again, in bold, and remember in my cases, in the materials I present, bold statements, underlined statements are good places for a test question. Results or tests are only as reliable as the attention given to each and every step of a procedure. You start cutting corners, you start cutting time, you will not get your accurate results. And then apply always quality control. Some items, when you get to chemistry analyzers, there's a quality control step that has to be done. And in microbiology, you learn with known items first before you look at an unknown, so that you can understand what you should be looking at. And always keep accurate records, whether this is in your clinical or practice drawing and taking great notes of your own lab work. And even in the next few weeks, I recommend that you practice drawing some of the items that you will be looking at, that when you come to the hands-on time, you will already have a little practice of that. So practice taking some great notes as if you were looking at an item as if it was a real patient. Coming into now the microbiology side, that was just lab safety. Take a quick little breather here for 1 second. Okay, so microorganisms. So we have 4 major sections, okay? We have, I'll start on the right-hand side of the slide, protists. Okay, these are like your amoebas. These are generally one-celled animals, okay? Or a cellular structure, one cell, that will have reproduction and so forth. You have fungi, okay? Molds, dermatophytes fit under that. Yeasts also kind of fit into the structure closer to fungi than bacteria. Bacteria is another one, and then viruses. So with viruses, as if we haven't heard enough about that in the last few weeks, but with viruses, they are either DNA viruses or RNA, and they are enveloped viruses. So again, think about some of your animal diseases parts and how that would approach this information. This is a need to know for the test or a test. Don't forget it when you come into the fall term for the hands-on time either. This is not in Unit 7, but you need to know this. And I believe that it's probably introduced in animal diseases, or it may also be mentioned in the section of in surgical skills. But it's called Koch's postulates. Robert Koch, around 1890, created this postulate. And it is used to today. I've even heard with the COVID-19, I've even heard some people discussing this. So it is current information on 150 years down the road. Koch's postulates. You should remember Robert Koch, probably 1890 would be a good date, and these 4 steps. The 4 steps are that a given organism has to be or must be present in every case of a given disease. So if you say that an animal has Pasteurella, then that Pasteurella bacteria needs to be present there in any of that disease. And that microorganism can be isolated from a specimen that's associated with the disease state, whether that is a fungi, a protist, a bacteria, or a virus. That microorganism can be isolated. And then you inoculate from that isolate into other susceptible animals, and they produce the same similar diseases, or disease signs, okay? And then that same organism associated with the disease state can be recovered from the representative specimen of the experimentally infected animal. So learn these 4 things. The given organism must be present. It can be isolated. It can be inoculated into another animal or susceptible organism. And that same organism can then again be recovered and prove that it is that organism causing the problem. Koch's postulate. You need to know that for the test. Microbiology. And I've tried to put some links to some of the pages, although there are times where the slides and the pages don't exactly flow totally. But again, this week you will be reading all of Chapter 1, or all of Unit 1 on the lab safety, as we've just gone over for 5 chapters, and all of Unit 7. Okay, because all of the PowerPoints that are presented this week support all of Unit 7. And then next week, you will come back to understand and put some of the foundations into place as you look at various organisms. So you'll be referring back into Chapter 7. Definition of microbiology, again, bold, you should write this down. You should be able to mention this back on a test. Is defined as the study of microbes, which are organisms that cannot be seen with the unaided eye. Therefore, microscopes are involved, etc. Careful attention to your quality control, your collection and techniques, and so forth. We might say that an animal or an organism has non-pathogenic organisms present. That's normal flora. In cattle, in rabbits, you could isolate normal bacteria from their gut, and it's not going to be causing a problem. But if that bacteria gets into the abdomen because of a wound, it could cause a problem. So non-pathogenic means where it's at, it's not being a problem. Normal flora being present. In fact, in some cases, we want good bacteria there. Bacteria is what's called prokaryotic cells. So read about that in the textbook. If you need to get further just understanding of prokaryotic versus non-karyotic, prokaryotic, you can Google that also on the web. And you're going to consider specific requirements of the potential specimens in collection and in preparation. You aren't going to prepare every single organism or possible organism in the same way. So various concepts to understand here. So this is actually slide number 14. My slide numbers that are in the corner came from a couple different renditions of putting these slides together. But I've tried to actually organize this layout to the textbook as best as I could. But concepts to understand about bacteria. Some of them have what's called capsules or flagella that could be present. Okay, not all of them have it. You can have some that are obligate aerobes. They need to have air present in order to grow. But then you have obligate anaerobes, okay, that really don't want much oxygen present. And then you have facultative or aerobes that do better with some oxygen, but can also do a little bit less. And then you have what's called aerophilic bacteria. These are supposed to be down arrows here on all of these where there is reduced oxygen tension present. And a lot of this is going to make more sense as you understand how some of these concepts form over the next weeks in skills and in various tests that we do. Capnophilic. They like to have an increase of CO2. So microaerophilic, they like to have decreased oxygen tension, but capnophilic wants to have actually an increase of CO2. So how can we do that? We put the specimen on a media plate inside of a little jar with a candle. We call it a candle jar. We light the candle, put the lid on it, and the candle goes out. What did we just create? A little bit of a increased CO2 in that candle jar, and then it is incubated, and those bacteria inside that are on the plate inside that jar are capnophilic. It is an increased CO2 area. Fastidious microbes. Some are just, they won't grow on just everything. They're fastidious, okay? We have what's called mesophils. They like to grow at 20 degrees to 40 degrees centigrade. So they kind of that middle ground, okay? And we have psychrophils, which want a decreased temperature, and thermophils want increased temperature. So I can see some of these arrows are a little bit confusing. So psychrophils, decreased. Thermophils want a little higher temperature. So if you have a specimen from a reptile that is going to be cold-blooded, you don't want your incubator at 37 degrees. You're going to want it less because they're psychrophils. When you look at identification of bacteria, you look for the criteria of the size of them, the shape, the arrangement, and the chemical reactivity. So all of those are very important. Match this up to the textbook as you look at it. When we talk about morphology, so the morphology of bacteria, okay, so we have three basic shapes, which are cocci, basically round circles. We have rods, or they're called bacilli. They're more of a rectangle. And then you have spirals, and most spirals are really bad bacteria, okay? You have some that are pleomorphic. They take a variety of shapes. They're kind of cocci. They're kind of a rod. They're pleomorphic, okay? You then can have singles of these. You might have a single cocci, or you might have a double diplococci, D-I-P-L-O, diplococci, or pairs, or you have a cluster, and we say it looks like a bunch of grapes because it's 10 cocci together, looking like a bunch of grapes, or they might be a chain. We also have some that are called palisades, and if you've ever seen palisades on a rock formation, they kind of form together, and we have some bacteria that take those formations and other arrangements. In your textbook around page 237, it talks about spores, and not all spores are identical. Spores are intracellular refractile bodies. So you should know that definition. I didn't underline it all, but you should know the definition of spores. Intracellular refractile bodies, and they're called endospores or spores, inside of, okay? They're refractile. When you use your scope and you go up and down out of the focus, you see them refract. What is the size of the spore? Where is the shape? And the location. Again, look at the textbook and notice the illustrations of those. So they might be a large spore, it might be a smaller spore. It might be sort of oblongish, or it might be round. It may be in the middle of the cell or toward an end. There might be two of them, okay? Usually seen as non-staining bodies when you do your gram stain. And they're resistant to heat, pressure, and chemicals, and that's why bacteria that have spores can actually live in the environment much longer periods of time, or kind of go dormant because they're actually resistant. The spore helps them be resistant. So understand the basic shapes: cocci, rods, or spirals, and understand about spores. Okay, bacteria reproduction, page 238. All bacteria are a single DNA strand. Okay, they reproduce what's called binary fission, and there's four phases of growth. You need to know these. This is a test question. I might ask you to put them in order. I might ask you to list them. I might say which stage is missing on a multiple choice question. Okay, so 4 phases of growth. I might say, which is the first one on a multiple choice question? The first stage is what's called the initial phase or lag phase. So look at page 238. You'll see a diagram about this. I usually draw it on the board, but I think your textbook can support the material. And then you have what's called the exponential phase. They double. This is also what's called the generation time. How long does it take for a reproducing or doubling of that bacteria being present? Exponential. And then there's kind of a stationary phase. There's no change or very little change. And then usually more of a logarithmic, faster decline. So understand these 4 phases of growth as outlined on page 238. What kind of equipment might you use? And even just in a general practice, basic veterinary practice. I'm not talking about a veterinary diagnostic lab. I'm actually talking more about the Aztec Animal Hospital, or even a VCA animal hospital. What might they have present? If you have a gram staining kit, new methylene blue stain, so new methylene blue is a stain. You want new slides. You need to have new slides to have reliable samples that you're producing. You'll have slide holders being present, transport media, Sterile rayon swabs versus cotton swabs. One of my other slides talk about that a little bit. And you might have some kind of you can use a lighter to heat fix. You actually heat fix the slides. So you might use a lighter or even a coffee warmer. Just a little electric coffee warmer can be fine. Or if you have, like we have in our laboratory, as you would see on the bench equipment video, we have a slide warmer tray. And then hazard containers. Steps that you might do. So there's not a whole lot of money that's involved in some of this basic equipment. But you might do a direct smear. You could look if it's gram positive or gram negative. Methylene blue is often used for fungi, maybe even yeast possibly. Streak isolation tests, in-house tests, and sensitivity tests. Coming down toward the end here. Okay, so stains. So in the textbook are many, many different stains mentioned. For probably test purposes, you definitely need to know a lot about gram stain. And you should know that new methylene blue stain is used for fungi or dermatophytes. It is also called lactophenol cotton blue stain. You should know that name goes together with new methylene blue. The Ziehl-Neelsen stain, acid-fast stain, you will not be using it in our laboratory. Usually that would be more in a diagnostic lab. You might use the GIMSA, G-I-E-M-S-A stain, usually, but it's again is typically for some different kinds of bacteria than what you will see in your laboratory work. In bold, know this one for a test question. All bacteria that contain capsules are pathogenic, but not all pathogenic bacteria contain capsules. So what that means is if you see a capsule, it is pathogenic. But a lot of other bacteria may be pathogenic that do not have capsules. Okay, so that's how that statement goes. And then there's flagella stains, motility tests. You'll be actually doing some of those when we come to the hands-on. I made a couple separate video clips that you should review about the gram staining. And this slide is pretty much brought up on in that context. Okay, but you need to know about gram stain. So look at your textbook and understand the different terminology. It is because of the cell wall composition that the gram stain test even works. Okay, and it is regarding the peptidoglycan matrix. Peptidoglycan matrix is what makes a cell be gram positive. It is a very thick cell wall. Gram negative has a thinner cell wall, a very almost non-existent peptidoglycan layer, but the outer membrane is what's called a phospholipid bilayer. So you need to know what makes up the cell wall of the gram stain peptidoglycan matrix, and a gram negative is the outer membrane is phospholipid. And it's because of that membrane that the gram stain process works. You need to know for the test that primary stain is that the first stage is called a primary stain, crystal violet. It's purple. The second stage is called a mordant. It fixes that stain into the cell wall. That's grams iodine. And then you have a decolorizer, either 95% ethanol or an acetone base. It's basically a clear liquid. It is the trickiest part to do a perfect gram stain because you can over decolorize very easily. And then the last stage is a counter stain called safranin. So look at the pages and also look at the videos that I have put together to try to help you to know ahead of time how to do the gram stain. So gram positive, the organisms look purplish. There's a thicker cell wall. It holds on to that purple stain. It holds that purple stain in place by the mordant, and with the decolorizer, it still holds it. It does not let it go, and it's due to the peptidoglycan layer. Gram-negative, thinner cell wall, gets the purple stain on there, but then with the decolorizer, it's released. It's gone, okay? And then you stain it with the counter stain of the safranin on that thinner cell wall. So understand the parts of the gram stain, the names of those gram stain. Those will be test questions in both primary stain, crystal violet, the mordant being grams iodine, decolorizer, and then the counter stain being the safranin. With media, there are 6 general types of media. One that you will be using the very first day when we have our hands on will be transport media, but you'll probably also be introduced to some of the others because of the little bit of speed up in this course. General purpose media, you have what's called enriched media, selective media, differential media, and enrichment media. Look at your textbooks between 243 and 246 to bring this into understanding. Enriched media is formulated media. It is the media, the presence in your petri dish is meant to meet the requirements of most fastidious pathogens, okay? Enrichment media are liquid media that favor the growth of particular group of organisms, and a culturette is used for specimen collection. Some of the general media that you will be utilizing through your time in the microbiology lab is, first one, blood agar. We look at what's called hemolysis patterns. Again, look at your textbook for what I mean by alpha, beta, gamma, and delta. Look at the pictures and how that hemolysis works. I always think about it like the bacteria is eating up the blood in that agar. You have McConkey agar, or we call it MAC, M-A-C. That contains crystal violet in it, and that will suppress, not 100%, but it usually suppresses gram positive, which means you'll use MAC on gram negative to get a better growth. And we have general nutrient agar, a basic media that you'll be using. The thioglycolate broth is for anaerobic bacteria. You won't really be using that, but you should understand it from the textbook. And again, urea tubes and slants, and what we call the SIM, sulfide indole multitubes. They're really cool if you ever have an organism that grows in one of them. They leave little black trails. SIM. You also have Simmons citrate tubes, triple sugar iron agar, brain heart infusion broth, mannitol salt agar, mannitol. It is selective for Staphylococci, especially it will isolate a Staph aureus. Again, we do not really have that in our lab, but you should probably understand that mannitol is selective for Staphylococci. And then we have bismuth sulfite agar, Mueller-Hinton. You will be utilizing that for an antibiotic sensitivity disc test, Mueller-Hinton. And Saburod dextrose, that is the type of media that is inside of a DTM tube for growing dermatophytes. And the last one is your yeast media. So that is microbiology PowerPoint number 1. Match it up. Definitely with my PowerPoints, remember things that are underlined, in bold, things that say this can be a test question, need to know this. All of those things are hints to understand the material. Some of this PowerPoint is actually going to be augmented with the videos, and enjoy reading the textbook, matching it up, and then you'll be looking at the chapter Microbiology number 2. Thank you very much.